Hey everyone, so I'm Maddie Thompson, and I will be going over my thesis today, which is assessing the toxicological effects of motor oil and tire particles on zebrafish. I have a lot to talk about today, so I'm going to go ahead and get started. So in 2021, Washington State Department of Ecology was funded for several projects regarding stormwater concerns. Stormwater is a growing problem because of increased urbanization. So pollutants from roads, industrial sites, and urban areas can enter waterways via runoff and become toxic to aquatic ecosystems. So with that being said, there are a lot of laws that govern stormwater management and water quality. And so under this funding, some collaborators such as Washington State University, the Research and Extension Center where my project takes place, was prioritized with the main topic of identifying areas of concern and then key contaminants with the main goal to prevent toxicity and reduce it overall. So for my collaborator, the main goal and identification for the contaminants with the main purpose of identifying how they affect aquatic ecosystems. So one way to do that is through the practice of environmental toxicology, which is used for assessing influences of contaminants. There are three major parts to this. So there's the environment, the chemicals, and the organisms. So for my project specifically, the environment is areas of increased urbanization, which are causing an influx of stormwater pollutants. The chemicals I will be using is synthetic stormwater. So one way to analyze the effects of stormwater on in a lab setting is by creating synthetic stormwater. So synthetic stormwater simulates chemicals that are found in runoff. So things like motor oil, tire particles, and other leachable chemicals. And then one way to test that is by using a model organism, zebrafish. So zebrafish are used for a variety of reasons in environmental toxicology studies. This is primarily due to their rapid development. So they are fully developed at 48 hours post-fertilization, and they have many genetic similarities to vertebrates such as humans and many other native species like salmon. And they also have transparent embryos. So you can analyze both genetic and abnormalities within morphology. One way to use genetics is by the path of epigenetics. So when an organism exposed to stress or environmental toxins, they produce a biological response. So genes are either turned on, meaning upregulated, or they are turned off, downregulated. So for zebrafish, they have a gene called the CYP1A gene. So this gene is associated and involved in the detoxification of foreign chemicals for vertebrates. So it is a biomarker that is used to track environmental pollutants, and you could use that in correlation to chemical levels. So taking this idea to needing to address how these pollutants affect aquatic organisms, and then using synthetic stormwater, for my case, motor oil and tire particles, and then applying it to the model organism to basically inform policy and regulatory decisions regarding stormwater concerns. So which pollutants are of higher concern? Which led me to ask the question, what are the effects of synthetic stormwater on the expression of the CYP1A gene in zebrafish? Again, to inform policy and regulatory decisions regarding stormwater practices. So my project has two main parts. So the first part is the toxicity exposure itself. This was a 48-hour zebrafish embryo exposure, which relied on spawning to the zebrafish in-house, sorting thousands of embryos, creating sublethal ranges and stock solutions for both used motor oil and tire particles, and then computing the exposure itself, which was a 24-hour water change where I performed water changes on all 6 of the different treatments for both tire particles and used motor oil. And then at 48 hours was the end of the exposure, where I took down the exposure and performed morphological assessments briefly, and then got them ready for their next step, which was measuring the biological response. So measuring the CYP1A gene. This was a very extensive process. So it was RNA extractions. So getting the tissues ready to be processed for RNA. And then from there, I did DNA synthesis, and then got them ready for qPCR. So actually measuring the expression of that biological response. There are not a lot of pictures, however, I do have this one. So this is the actual PCR product, what you will be putting into the instrumentation to measure the results. Each tiny dot at the bottom of that qPCR 96 well plate, it represents 40 individuals. I ran that 96 well plate 4 times, so 96 times 40 fish times 4. It's a lot of embryos. But that is a great picture, and I feel like it needs to be highlighted. So here's some other pictures of me doing the work. There is the WAF that I created for used motor oil, some zebrafish, performing some analyses on morphology, and then actually the instrument that I used to measure. There are no pictures of the actual DNA extractions. However, that's because it's used in a controlled setting, no sterile, very sterile environment. So let's put that together. So we have this exposure and the biological response, and I was able to compute the mean gene expression across treatments. So this graph here, so we have the control on the first side of the x-axis, and then leading to the highest concentration. And then the y-axis represents the gene expression, so the biological response of the CYP1A gene. So the capital letters here denote significant differences among treatments. Treatment for the control is indicated by an A. No other treatment has that because it's significantly different. However, if you look at treatment 1 and 2, so our lowest concentration next to the highest, there's some similarities indicated by a B, and then C for the 2 and 3, 3 and 4 indicated by a D, however, and 5 included, which is great. So the main takeaway here is that a higher concentrations resulted in a higher biological stress response, meaning that they were producing more of the CYP1A gene with an increase to exposure. Similarly, for the tire particles, this is the mean gene expression across the treatments. So control indicated A is significantly different across all treatments. Treatment 5 was my highest concentration, and it resulted in total mortality, so it wasn't included on this study. And again, that was 10 grams per liter for tire particles. However, for this gene expression levels of CYP1A gene were much lower for this. I don't remember if you remember this. This is 1500 versus very low 200, representing how much of that CYP1A gene is expressed. Still, we do see a higher concentration resulting in a higher biological response. So what does that really mean? So for the used motor oil, there was indications that it is a stronger defense for the CYP1A gene when exposed to all treatments. There was a dose-dependent increase between CYP1A gene across all treatments, and then there was some overlap across treatments 1, 2, 3, 4, and 5, which is also good because it means we're targeting a range and encompassing a range that there shows overlap to where they're close enough together that there's probably not much difference if you were to include ranges in between there. And then the highest concentration didn't result in lethality, which is great. For tire particles, however, there was a much weaker defense. So a lower higher concentrations still resulted in a low dose response for the CYP1A gene. The treatments themselves didn't differ significantly, and then the highest concentration resulted in total mortality. But, so neither treatment really incorporated the lower sublethal range because the control treatment was significantly different than the first treatment. If there would have been some overlap between the control and the first treatment, that would have been great, so we would have known when the CYP1A is first induced. But, so this study does still really highlight the potential impact of these contaminants on aquatic ecosystems, and it does highlight the significant threat that, the increase of used motor oil and tire particles and their presence in urban ecosystems, and then in turn entering into streams via stormwater. We are acknowledging, though, that this study doesn't accurately reflect environmental conditions. So there's a lot of more complex interactions that are occurring when tire particles and motor oil are being influxed via stormwater. But we still can ask questions further from this study to when is it first induced? When is CYP1A first induced? And then if we were to perform morphological assessments simultaneously, so having this study and then another study comparing morphological abnormalities to quantify how much that gene is being expressed and at what degree are we seeing pericardial edemas, yolk sac edemas, scoliosis, and things like that. So that's pretty much the scope of my thesis project. With that being said, I would like to thank my collaborators. Aaron Martin, this project wouldn't have been able to happen without you, and I'm very appreciative of that. Carrie for carrying this project through and supporting me, and then Dr. Jen McIntyre and Stephanie Blair for just being endless support and great partners on this collaboration. So are there any questions?